I assume my presentation is going to come up soon. Okay, so I am a conservation geneticist. And so obviously I'm a strong advocate for using genetics to inform conservation efforts. And so what I'm going to do today is really just kind of try to give you a broad overview of conservation genetics and then kind of highlight some areas and some information where you can really get good information for conservation using genetics. So what I'll do is I'll first start out with a brief overview of conservation genetics, and then I'll move into several examples of how genetic data can be used for conservation. So for example, using genetics to identify clonality, and then the number of individuals per site, using genetics to identify the mating system, genetic diversity and structure, and then the conservation implications, both for in situ and ex situ conservation. And then finally, using genetics to identify populations that can be most susceptible to climate change. So next slide. Okay, so as you guys are probably well aware, biologists recognize 3 fundamental levels of biodiversity. There's ecosystem diversity, species diversity, which I think is kind of the main dimension of biodiversity you guys have been thinking about in these sessions. And then finally, there's genetic diversity, which is kind of the level of diversity that I think most about. So next slide, please. So genetic diversity is important because it produces the physical and functional differences among individuals in a population. And it promotes resilience and adaptive potential. And that's really important in the species where there may only be, for example, like 30 or 50 individuals left. We want to make sure that we have as much resilience and adaptive potential as possible. So next slide. But I would argue that our knowledge of biodiversity is incomplete. And I think there's been a lot of talks that have talked about that at the species level, where species could be taxonomically questionable, there could be hybridization, there may be cryptic species out there. But it's particularly so at the level of genetic diversity. And this is because genetic diversity is just not something you can readily observe, as Joyce has pointed out. And it can be affected by all kinds of things that you really don't know. For example, there could be a genetic bottleneck that has reduced genetic diversity in a population. There's different life history traits that can shape the genetic diversity of species or populations that we aren't aware of. So next slide. So this is really where I think conservation genetics comes in, because I don't think, we can't really do a good job of conserving biodiversity if we don't know what we have. And so that's where I think conservation genetics comes in. It's a field where we use genetic data to quantify both species and genetic diversity and make sure it's conserved. So next slide. Okay, so in my lab, the way that things generally work is that I work with partners, especially like people in agencies such as yourselves that are responsible for conserving specific species. And the next slide. And a lot of times they will come to me with specific questions that they know that can be answered with genetic data. And there'll be questions like, for example, is a species clonal and how many individuals do we actually have? What are the mating system and life history characteristics of the species, which can be answered with genetic data? And what are the consequences for genetic diversity, genetic structure, and conservation? Have populations experienced declines in genetic diversity? There's things that are factors that can be associated with small population size, including genetic drift, genetic bottlenecks, and inbreeding that can reduce genetic diversity. And then finally, one of the newer applications for genetic data is trying to identify populations that may be susceptible to climate change. And these are really questions that are kind of focused on in situ conservation, so conservation within natural populations. But next. There's also a bunch of questions that can be answered for ex situ conservation, which Joyce kind of touched on quite a bit, and I'm not going to be talking about much today. But For example, we can see how much genetic diversity is present in ex situ collections relative to wild populations. And this is particularly important when you have these really rare species and you want to make sure you're getting every single individual into an ex situ population. And then also how we can design reintroductions to make sure that they have high genetic diversity. Next slide. Okay, so I know you guys have gone over some genotyping techniques, but the ones used for population genetics are slightly different. So I'm just going to talk about how these things differ for population genetics. One of the things that kind of differs is that we collect quite extensively. And as Joyce said, we try not to do any harm to individuals in the population, but we will collect usually leaf tissue from a large number of individuals. Usually a minimum of 10 if that's the population size that you have, 24, or sometimes we'll collect samples from every single individual in the population depending on how rare a species is. And then we'll try to collect from as many populations as we can. And that's usually for in situ questions. But in ex situ, it's the same thing. We'll go into a collection and we'll sample leaves from botanical garden collections or from seed banks. Next slide. And then we'll take those samples back to the lab. This is what my lab looks like next. And then we'll take the samples and we'll extract DNA out of them. And so we'll, a lot of times, depending on the rarity of the species, we could have hundreds of samples. Next. And then we essentially take those the DNAs, and we genotype the same highly variable DNA regions from different individuals and compare them. Next slide. So there's two types of markers that I think are most commonly used for population genetics. And I think you'll probably see quite a bit of these in the kind of the future talks that are after mine. The first is microsatellites, and the second is Rad-seq. And so next. So I'm just going to go over this really briefly, just so you guys have an understanding of the markers we use. So these are DNA repeats, microsatellites. So it's essentially like a CA, 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 CA. And the way that we identify these is that we'll sequence genomic DNA, and then we'll design primers along the flanking regions. And then we'll take those and we'll test them in a variety of individuals. And then we'll get a panel of microsatellites, usually somewhere between 10 and 40. And then what we'll do is we'll do a PCR of each marker on the DNAs, and then genotype them on a capillary sequencer. And the markers will look something like this. So the first three on the right over there are homozygotes. And you can see there's three different alleles with the size of 235, 238, or 241. So those are each different alleles. And then the ones in the bottom are heterozygotes. And in general, microsatellites, we use a much smaller number of loci. It'll be like, 20, but they're very highly variable, so they're very informative. And there's some time involved in scoring the data, but the data are they're very clear. So next. One of the things that microsatellites can be really good for is looking at variation in ploidy. So, we have diploids or tetraploids, for example, and a lot of times tetraploids will have like 4 alleles per locus. And so that can kind of illustrate differences among individuals in ploidy across populations or within populations. And then these are markers that can be used for genetic diversity, genetic structure, inbreeding, looking at the genetic similarity of populations, and then also identifying, interspecific hybrids like F1 hybrids. Next slide. Okay, the other type of marker is Rad-seq or GBS. So Rad-seq stands for restriction associated DNA sequencing, and GBS is genotyping by sequencing. And this is an approach where you take high quality DNA and then you digest them using restriction enzymes. And then it usually involves some sort of a ligation of adapters onto the sticky ends left by the restriction enzyme. And then you usually do some sort of sample specific barcode addition through PCR. And then you pool the samples and sequence it. And we'll usually pool 96 individuals and put them on a Illumina High-seq. And it will bring back about 400 million sequences. So because of the sheer number of sequences, the analysis of this type of data is a little bit more complicated. But one of the great things about it is you'll get like, you know, it can be up to tens of thousands of loci across the genome of an individual. So we get a lot more variable, not more loci, but they're a little bit less variable. And next slide. This is suitable for all the things that I mentioned related to microsatellites, but it can also be used for phylogeny, and then also the analysis of susceptibility of specific populations to climate change. Next slide. Okay, so then once we have our genetic data, then we can analyze things like genetic diversity, so looking at allelic diversity, whether populations have unique alleles that are present nowhere else. So for example, they're called private alleles, and those may be populations that you want to specifically protect that specific variation. And then we'll look at heterozygosity, so kind of averaged across all the loci. And that can tell us a bit about inbreeding in populations. So if things are very homozygous, it can tell you if a population has been inbreeding or if a species has a self-mating system. And then we'll also look at genetic structure. So in the lower right-hand corner is a structure diagram. So this is essentially those blocks of colors represent groups of individuals that were sampled within a specific site. The vertical lines represent individuals. The colors represent groups in which individuals are assigned. And the way this program works is that essentially it shuffles individuals into different groups and tries to maximize Hardy-Weinberg equilibrium. And then it essentially tries to find an optimal grouping of individuals into genetic clusters. And this is a really important tool for us. And essentially then what you can do is you can use this information to say, well, let's try to protect a population that has all of a population from each one of these genetic groups. So each one of these colors. And usually they're spatially structured. So you might say, okay, we'll protect one of the blue populations and we'll protect one of the pink populations. And it's kind of a way to make sure that all of the genetic structure and genetic variation is conserved. And in terms of ex situ, then we want to make sure that we're collecting from each one of these groups and have a representation from each one of those. All right, next slide. Okay, so I'm just going to show you some examples of how we've applied this data to help guide conservation decisions for endangered species. All right, next slide. Okay, so clonality is a really common thing in plants. There's like a whole suite of plants that are clonal, and it's essentially where you just get individuals that will have rhizomes that bud and they form a new genetic individual that's usually right next to the parent individual. And essentially what happens is you get a group of neighboring individuals that when you go out and genotype them, they're all genetically identical to each other. But what it means for conservation is that generally you may have a lot fewer individuals in a species than you like previously thought. And another thing is that a lot of the species out there that are clonal are also self-incompatible, which means they have a genetic system of self-incompatibility that prevents them from self-fertilizing. They cannot produce fruit. And so there's a lot of species out there that end up, you know, they're very rare and they have attrition of genotypes over time. And they may get to the point where there's really no cross-compatible mates in the population. And in that case, it's kind of a situation where you almost get like a collapse of a self-sustaining population. So next slide. So there's an example of this that I've been working on for the past, 15 years. This is Ziziphus celata. It's this long-lived shrub that's found in 3 counties in Florida. It's federally endangered and it's known from 16 sites. And this species has been declining for years due to habitat loss, and it's naturally rare. So next slide. So this species, when you go to a population, it looks like it could be a healthy population, but when you actually go out and genotype it, it turns out that you know, so in total there's about 1,000 things that look like individual plants, but when we genotype them, it turns out that we've identified only 43 genotypes. So even though it looks like there's thousands of individuals, there's actually only 43. And not only that, there's only, of the 16 populations, 11 of them contain only a single genetic individual, and then 5 of them contain more than one genetic individual. Next slide. So this is one of those species that has a genetic system of self-incompatibility, and there's been some cross-pollination experiments, and they've identified only 2 mating types in this species. And it turns out that in every single population, there's only a single mating type. So essentially, there's no natural sexual reproduction occurring in any population of the species. And there's also no natural seed germination or seedling recruitment occurring. So next. So it's a situation like this where something is this rare, where you might have only a dozen individuals or 15 individuals. They're really good candidates for kind of more extensive ex situ conservation efforts. So in this species, there's been efforts to take clones of all of the individuals of the wild genotypes, and they've been put into a botanical garden. And then because there's multiple cross-compatible genotypes that are there, they're actually like producing fruits, and those fruits are being germinated and then put out for reintroductions. But it's really important in the situation where you have these reproductive issues to have genetic information to guide it, because otherwise you would just be working blind. Next slide. Okay, so so generally, kind of the point here is that the conservation picture really differs if you think you have 1,000 individuals and you actually only have 30, right? The species just becomes a lot more rare. And not only that, like these species that are highly clonal a lot of times have some pretty serious reproductive issues. And I think this kind of underlines what Joyce is saying, which is these really rare species where there's very few individuals are really good candidates for genetic analysis. And they're also really good candidates for ones where you want to do some more extensive ex situ conservation and maybe reintroductions as well. All right, next slide. Okay, next I'm going to show a series of studies where we've used genetics to identify mating system, genetic diversity and structure, and then conservation implications. All right, next slide, please. Okay, so what I'm going to talk about now is essentially kind of 3 midwestern annual species where we've used genetics to try to devise conservation strategies. So all of these species are federally threatened, and they have a lot of similar aspects to their biology, in that they're all endemic to rock outcrops or glades in the Midwestern U.S. And because their natural habitat is fragmented, they have a naturally fragmented island-like distribution, but they're threatened by habitat loss and fire suppression. And they're all winter annuals, and we didn't know the reproductive biology when we went into the studies, but we did know that their seeds were gravity or water dispersed. Next slide. So all of these studies were funded by the Fish and Wildlife Service, and essentially what we did, the research questions were similar. They wanted to know how genetic variation is structured within and among populations, and tried to use the data to devise a strategy to conserve the genetic variation. Next slide. And actually, I should mention before I get started that because the biology of these organisms were so similar, we were really expecting to find similar patterns of genetic structure. So this is the first one. This is Leavenworthia exigua, and this species has 2 varieties. Varlicineata is this variety that's found just south of the Louisville, Kentucky metropolitan area, and it's federally threatened because of the extensive habitat loss due to the kind of urban sprawl that's happening there. And then there's also a variety exigua, which is found in Tennessee. Next slide. So what we did basically is we went out, and I think we sampled from about 20 populations, and we sampled 24 individuals per population. And we sampled from both variety Laciniata and from variety Exigua. And so for this, this is a genetic distance tree. So the way that you interpret these is, so each one of those branches represents a population. And so there's like 24 individuals that we sampled within that population. And then the branches are proportional to the similarity among populations. So populations that are more close to each other are more similar. So as you can see in variety Exigua, you see relatively large branch lengths separating the populations. And essentially what we find is that the populations are somewhat different from each other. But in variety Laciniata, what we found is essentially that every single individual in this species is genetically identical to each other. So they essentially have a fixed genotype that is found in every single individual. And next slide. And we think that what's happening here is their program, and we're trying to capture all the genetic variation in the species. But it turns out if you want to capture the genetic variation, you really only have to collect seeds from a single population, captured all the genetic diversity. And the same can be said for the in situ. You really just need to protect a single population. And so in this species, I mean, I think it's better to maintain more populations, but if your sole goal is to protect genetic diversity, you don't really have to do very much. Next slide. The next species is Geocarpon, and this is this tiny little thing. That plant up in the upper left is like the size of a dime. And I think Mike knows this one too, because it's a Caryophyllaceae. Anyhow, so this is a species that occurs throughout Missouri, Arkansas, and then there's a few populations in Texas and Louisiana. And it's a habitat specialist, and in the northern part of its range, it occupies sandstone glades, and in the southern part of its range, it occupies a habitat called saline barrens. And so we were expecting maybe genetic variation to be structured by these different habitat types. So for this species, once again, we went and we tried to sample from throughout the range of the species. At each one of those sites, we sampled about 25 individuals, took them back and genotyped them, and looked at patterns of genetic structure. And next slide. And in this one, what we found is essentially really, really strong structuring of genetic variation. And in fact, the reason why this is the case is because the species shows all the indications of being sulfur. So essentially, each population is extremely, extremely homozygous. Almost zero heterozygosity is found in this species. So essentially the way this works is that an individual like self-fertilizes and essentially drops genetically identical seed right next to each other. But in selfing organisms, there's really strong genetic drift. So you can get very strong divergences among these selfing lineages. And so the reason why we see such strong genetic structure is because each of these populations represents a selfing lineage. And so in order to protect the genetic variation in this species, what you have to do is you have to protect every single site that the species occurs in, which is not, if you want to protect them in situ. So obviously that's not very feasible. And so for this species, it's a really good candidate for protecting genetic variation in ex situ collections. Next slide. Okay, and then the last species that I'm going to talk about is Physaria filiformis. And this is a species that occurs southwestern Missouri, northwestern Arkansas, north central Arkansas, and the Ouachita Mountains. And it's got this patchy distribution that corresponds to the presence of suitable glade habitats. So for this one, once again, we went out and sampled populations at each one of these sites and looked at patterns of genetic structure. Next slide. So for this one, what we found is that okay, so for example, the three southern populations you see on the left-hand slide in yellow, blue, and purple, those correspond to those three major geographic groups. And then within southwestern Missouri, which you see on the right, you see that there's a relatively strong structuring of genetic variation according to location. And so this species is self-incompatible, like I talked about earlier. And it's also insect pollinated. And so essentially what we're seeing is this genetic structure according to kind of the distances that insect pollinators can travel. And so in order to protect the genetic variation or conserve the genetic variation in this species, what you need to do is protect a population in each one of these genetic groups. Next slide. And in fact, in this species, the only set of populations that doesn't have a protected site within that group is these ones right here. And so those are the priorities for kind of public protection because it contains a unique subset of genetic variation. So, so basically in those three examples, essentially what I've shown you is that even though we thought we were going to see these very, very similar patterns of genetic structure, what we have seen is that it can vary really widely, and we had no idea about that at the time. And so not only does genetic structure vary, but the kind of the strategy, the best strategy for protecting genetic variation in these species varies. And so I think it just highlights how important a genetic study is for these rare species. Okay, next slide. Okay, and another thing that we found, this is just kind of another little thing that we found in this Physaria study, which is the species I presented last. So once again, we have a genetic distance tree, and so populations that are more similar to each other are closer together. And what we found is that the populations that are kind of in the northern part of the species range, so those three like circles that are above the line on the lower right-hand map, they all cluster relatively closely together in the upper part of that figure on the left. But so next, please. But we also sampled an outgroup, and this is actually really useful for kind of rooting a tree. And what we found is that the populations, the Ouachita populations that are found in the southern part of the species range are actually as genetically divergent from the rest of what was supposed to be the same species as the outgroup. And it actually looks like they're more genetically similar to the outgroup. And actually, we've done some additional research, and we found that they occupy two different physiographic areas. They diverged about 15,000 years ago. They have a different petal morphology, and we're in the process of describing it as a new species, which actually only occurs in 4 sites, and I think there's less than 1,000 individuals. But you know, this is also something that we weren't expecting to find, but genetic data, like a genetic study like this, can also kind of highlight species boundaries as well. And then the other thing is that this has some important conservation implications. One is now we have a new highly endangered species, but the original range of the species that we were studying has now shrunk significantly, and there's a lot fewer individuals. So the conservation outlook on that species is a bit more dire as well. But the good thing is now that we know we have these 2 species and we can work to conserve them. All right, next slide. And then finally, I just wanted to highlight that this information can also be used to detect losses in genetic diversity. When you have small population sizes in endangered species, populations can be prone to things like increased inbreeding, genetic bottlenecks, and then losses in genetic diversity due to genetic drift. So these are kind of indications that populations may be at risk or they could be declining. And so it's kind of a way to measure population health or population resiliency. And so if we find that one of these processes is occurring, then we can maybe target that population for management or translocations or augmentations. So for this one, this is once again Physaria filiformis. We can look at observed and expected heterozygosity and compare those 2 metrics. And if we see much lower observed heterozygosity than expected heterozygosity, that indicates inbreeding. This species, generally, they're consistent across the populations. So we didn't detect inbreeding. And I should mention that inbreeding is only really a problem if a species is not naturally a selfer. So in this species, if we saw a lot of inbreeding, this would be a problem. But in a species like Geocarpon, which is kind of a natural selfer, it's not a big deal, right? So you have to kind of gauge based on kind of the evolutionary history and normal conditions for a species. But okay, so next slide. But so we can analyze computationally evidence for a bottleneck. And in this species, what we found is that this population that I highlighted in yellow experienced a genetic bottleneck. And we think this is actually has to do with management. This population was very small, and then actually recently had been managed and had a remarkable increase in population size. And so we think that's why we see evidence of a bottleneck. So there's really not anything to worry about in this situation. But next slide. But we do see over here, see these two populations over here on the left. You notice they have much lower genetic diversity than almost all the other populations out there. And we think that these are populations that are experiencing losses in genetic diversity due to genetic drift, which is essentially just a stochastic loss of diversity in the population over time due to small population sizes. And so these two populations are really important targets for future management. So they're experiencing declines. And so these are the ones that we've kind of highlighted to land managers to kind of think about implementing more intensive land management to try to increase population sizes. Next slide. Okay, so hopefully with all of these examples, I've shown you one that the most effective strategy for conserving genetic variation can really vary widely depending on the characteristics of a species. Like I said, we found 3 different species with 3 completely different strategies for conserving variation. And you really can only find out this information if you do a genetic study. Next. Not only that, the same genetic information can be used to, detect cryptic species out there that we didn't expect to find. And then we can also highlight populations that may have experienced declines in genetic diversity that could be targets for management. Next slide. So I just wanted to, obviously I'm a big proponent of genetic studies, but I do think that if you have a really rare species, a genetic study is absolutely essential for helping to devise conservation strategies. So next. Okay, finally, I just want to show a really brief example of how genomic data can be used to identify populations that are most susceptible to climate change. Next. Okay, so this is actually a study that was conducted by Gugger et al. And this is actually a Hawaiian example. So this is a more widespread species, but this is kind of a relatively new application of genomic data to kind of devising conservation strategies. And so I wanted to highlight it. Because in general, it seems like mostly the examples I found are in widespread species, but we think it can be applied to more rare species. And that's something that we're working on right now. But so this study, what they did is they went out and sampled heavily of populations of acacia across the Hawaiian Islands, but focusing most intensively on the Big Island. Next slide. And they went out and they genotyped all these individuals, and they looked at the association between environmental characteristics and genomic variation. And they found that the most strongly associated factor that structures genetic variation is precipitation. So they found that there's unique genetic groups that are adapted to the wet conditions found on the east side of the island. They then basically looked at projected future climate change, and they found that precipitation on the east side of the island is supposed to change dramatically under these future climate projections, and that the individual groups or the genetic groups that are found on the east side of the island will probably be mismatched between they'll be mismatched to their future environment, and they're the ones that will be kind of the most at risk in the future. And so using that information, they propose a series of conservation measures, management measures to try to protect those populations and to kind of devise conservation strategies. And I will mention that this is something that we've seen mostly in widespread species, but we're trying to apply it to an endangered species of hibiscus from the Hawaiian Islands as well, in collaboration with Jeremy Fant and Zoe Diaz-Martin and my PhD student. So this is something that we're working on, and hopefully we'll have some interesting results coming up in the next year or two. Okay, next slide, please. Okay, so in conclusion, hopefully I've shown why genetics are important for the conservation of biodiversity. And, you know, a lot of times we find these really unexpected results. You know, 9 times out of 10, I do a genetic analysis and I'm always floored by the results because they are always completely different than what I was expecting. And either, you know, either genetics are great or I'm just terrible at predicting what I'm going to find. I don't know. But anyhow, we really do find some important and unexpected things, and they can be really important and relevant for conservation. Next slide. So not only do we find these unexpected insights, but I think they really can provide a very clear framework for conservation, you know, and prioritization of populations. And then, for example, in really rare species, making sure that you are conserving every single individual. So I think they're really important. All right, and with that, I'd like to thank some of my colleagues that have been collaborators. Next slide. And thank you guys for listening.